talk about the story of my work, but I'm going to try to relate it to how it might fit in with teaching or how it might help people refine their questions to get what they really want to ask. So this is going to be my story. I am currently a staff scientist in the wall lab, like Donna said. Am I not on?
I'm not on.
Am I on now?
We can hear you.
I don't know if we need it.
Yeah, we need it for the video recording.
Okay, turn now.
Am I on now?
Yes.
All right. All right. Thank you. Thank you for having me. I am going to talk about the story of my work. This is a discovery that I'll explain that I made when I was in graduate school. I'm now a staff scientist in the Wall Laboratory where I've worked on not only what it is, but what it does. And hopefully next summer I'll be investigating this further in my own laboratory. So this is the whole genesis of a lab, essentially. Okay. So just some background. I study pancreatic cancer, but I study very early events in pancreatic cancer formation. And this is because it's a very deadly disease. It's currently the third leading cause of cancer-related deaths. There's this terrible 9% 5-year survival rate, but when I started in this field about 12 years ago, it was half this. So we have made some progress, although it's very slow. One of the saddest things is about 52% of patients have metastasis at the time of diagnosis, and this drops the 5-year survival rate to 3%. So basically, we don't have any diagnostics. And a lot of this stems from the fact that we don't know how it forms. There's actually a lot of parts of pancreas biology that we still don't understand. So it's really important for us to do this work and see how the pancreas actually progresses to this cancer so that we can detect it and treat it. One of the more interesting things about this cancer is that almost all of them have a mutation in this KRAS gene. You'll see some other cancers like lung cancer will have a lot of heterogeneity in what mutations cause the cancer. But for pancreas, it's almost always KRAS. And then just a little bit about pancreas biology. I think everyone here probably knows, but the endocrine pancreas makes hormones that help for digestion, like insulin and glucagon. But the exocrine pancreas is made up mostly of asner cells, and this makes digestive enzymes that help break down carbs, fat, protein. The thing is that the pancreas is located in what's called a retroperitoneal location, where it's really hard to get to. It's really hard to monitor. It's not like the breast or the colon where you can just check and see if there's a cancer. And this makes it very difficult to study. So the pancreas is largely made up of these cells. They're called asner cells, and they make these digestive enzymes. And it's largely thought that these cells are what's actually seeding the cancer. So here's an up-close look by Electron microscopy. You can see they're pretty cool structurally. So how does this happen? When these asner cells become injured, they undergo a process called metaplasia, where they become ductal. And you've probably heard of this term in the context of things like Barrett's esophagus or Barrett's metaplasia. And what that means is that the tissue lining the esophagus becomes intestines-like in response to injury. So cells become other kinds of cells to aid in healing. So in the pancreas, we think these asner cells become ductal, and it's from this stage that they can proliferate and heal and become functional asner cells again. But when you have a KRAS mutation, which I mentioned was the most common mutation driving pancreatic cancer, the cells never heal. Instead, they become more and more irregular until cancer develops. So when I was doing my doctoral work, I studied this process, and I spent a lot of time looking at these lesions, and I kept staining them for different proteins based on what my boss was interested in. Right? And so I found a lot of heterogeneity, and that was known in terms of what pathways might be on at a given time in a cell. But what I kept finding is that these cells had a very specific morphology. And
Can you say a little bit more about what you're seeing here?
Sure.
Because I don't see.
So this duct, this is really simplistic. They're not really circular. It's this duct.
Okay.
So this is what asner cells are becoming, all these different cells. And in green, we're looking at a signaling protein, and we were looking at that because that particular protein is a driver in cancer. And so we would expect to see some cells higher than others, but if you look at this, you can see that it's really specific pattern. And then if you look at the red, you can see there's red at the very top of the cell in every single one. So what this is pointing at is it's more of a cell type instead of a cell that's on or off. So people had known that these cells were all different in terms of what they had on or off, but not that they're actually physically different cells. And that's what I'll get to. So basically, I saw this, and I said, well, what is this? I thought it was cool. I spent a lot of time on a microscope looking at signaling pathways for someone else's project, and I saw this, and I said, this is really cool. And the first thing you kind of do here is you start with an observation. Most scientists that are successful are working on something that they care about, that they think is interesting, that they want to go to work to do, that they want to spend 1 million hours on, right? That they have something invested in. And it's also really important because you build this laboratory of people that are working for you but with you, and you want them to work as hard as they can, so you want them to be excited as well. It's all about motivating people to be interested in this topic. So I was really excited. I thought this was really cool. It's an observation. Is it interesting? Typically in biomedical research, people decide this based on whether or not it's going to have some relevance to human disease, right? That's most fundable. That's what government funding wants to see. Is this going to help people? But it's also really important to expand our knowledge, particularly for reasons I already mentioned, which is we don't know enough about the pancreas. So a lot of these basic discoveries we make are going to seed treatments down the road. So is this interesting and worthwhile? Yeah, because we don't know what it is. We don't know what happens in the pancreas. Is it driving tumorigenesis? So I found this in graduate school, and I said, what is it? So the next thing I did was I gathered information, because there was no way I was going to look at a picture of it and figure out what it was. So I just started doing really basic things, like how many are there? Where are they? How do they, or when do they form? So in this case, I looked at mice that had a KRAS mutation. They go through a series of lesions that get worse and worse until they get cancer. So I found, and these cells are marked here in brown in this case, that most of these cells were forming in very early disease, and they were lost as we got to cancer. So now I have a pattern. And this was consistent and reproducible. And then the next thing I did was I talked to an expert, and I can't emphasize this enough. I've read a number of blogs recently where people really want to move away from the idea of the lone genius, right? You think of any kind of really important discovery in history, and you think of one person, and it's usually a man, and that's just not true, right? They have laboratories, they have businesses full of people working on these problems, and you can't solve them by yourself. And I think science is becoming more and more collaborative. People are getting better about speaking about their science, and it's really the only way to develop these projects quickly. So what I did was I went and I talked to a pathologist, someone that studies the architecture of tissue, and I said, what is this? And he said, I don't know. But he said, here's a textbook full of pictures of different cell types and different organs. Cell types are generally thought of as differentiated. They're functional, like an eye cell, so you can see. Different cell types in the intestines secrete something specific. So these differentiated cell types have a structure and they have a function. So he gave me a book full of pictures of different kinds of cells and different kinds of organs. And what I did is I looked through the book, and I was pretty sure I saw these cells in almost every organ. So definitely take advantage of another person's expertise, and this will come up a few more times in this talk. So like I said, I thought I saw these cells in all kinds of organs, so I went through the mouse, I took out every GI organ, and I stained for these cells, and I found, they're here in brown, I found a ton of them in the gallbladder. So then you just go to Google and you say, what cell types are in the gallbladder? Well, there's a lot of tough cells. And so what you're looking at here, this is an electron micrograph. The cell is outlined here in red. And this is from pancreatic disease. We have a differentiated cell type. It can be identified by structure, microvilli, actin rootlets, vesicles, and all these mitochondria. But at the time, people still didn't know what they do, right? So now we know what it is, but we don't know what it does. So that was back in 2014. The function of those cells was not known. Yes?
So were the tough cells the one in your previous picture that were fluorescent green?
Yes. Let's see if I can get back to, just want to point one thing out here. I was studying these pathways that are activated, and I kept seeing, I don't know if you could see here, this double line. And we had no idea what this was. Is this like a lipid raft? Is this a, turns out this is the cell membrane, and also the tips of the microvilli are lighting up. So it makes sense now, but at the time, what kind of structure is a double membrane on the apical surface of a cell? No idea. But that's what this is here in green. So now the red, and I'll show close-up pictures. The red are those actin rootlets, and the green are the microvilli.
What are the red?
Actin rootlets.
Oh, yeah.
So you can see those, see a little closer up here, and then these actin rootlets. And those are interesting. I've talked to a lot of hair cell scientists, people who study the cochlea, and it's generally thought that the length of the microvilli corresponds to the length of the rootlet. So like this is a continuous structure, it's just that this part is sticking out of the cell. So cells with actin rootlets this long, no one knows what they do. They haven't really been identified before. So back in 2014, people had no idea what these cells did. And in 2016, several papers came out where people identified a function in the intestine. So that's some place to start. And this is about when I joined Dr. Wall's lab here, and I went from what is this to what does it do? So in the intestine, these tough cells are fighting infection. So they have protozoa, parasite infection. And they do this by, I'm not going to go into this too much, but by secreting different substances that communicate with the immune system. So that's interesting. We can see them communicating with the immune system. And this helps us refine the hypothesis to what they might be doing in pancreas, but it absolutely doesn't answer it.
So your cells are the brown ones with the little things sticking out?
Right here. But this, and they are different in the intestine.
So I think you start by saying that, like the epithelial cells of the intestines, when they get injured, that other cells come in to aid in that and assist, and then, so are these, is that right?
Different tissues will become different types of cells in injury. That is an interesting point. At least in parasite infection, these cells are very rare. When there's a parasite infection, there are signals to stem cells to make a ton of them. And then those are necessary to communicate to mucin-producing cells. And then the mucin-producing cells just excrete tons of mucin. And they call this a weeping effect. And this sweeps the parasite infection from the gut. So it's called a weep and sweep response. So you are right that the cell types that make up the intestine change in response to injury.
And then, so in this one, if it had some kind of injury with those after the repair and the weeping, with those, if it were the case of bacteria or whatever, microbes, would those go away or change back into something, the original ones that are like in blue there?
It's actually, so the blue cells are, here they would have stem cells, which they've labeled here in the crypt. These are enterocytes, so they absorb nutrients. These are just two examples of secretory cell types. The intestine has 5, and they all perform different functions. And then the intestine reacts to injury differently than the pancreas would. So there's a lot of plasticity in the pancreas, just normally. Normal tissue will become tough cells in these different cells and then heal. There's no known dedicated stem cell in the pancreas, but there is in the intestine. So if you have this massive injury in the intestine, a lot of these cells will just die, and stem cells will make more. Under conditions of mutation, there's a little more plasticity from normal cells if they're mutated, but if it's just an injury, most of the lining will just die. So that makes the pancreas pretty, I want to say, unique. Okay, so when I joined the Salk Institute, Dr. Wall and I were very interested in the idea of what do these cells do. So we asked a very basic, broad question first. Are they pro or anti-tumorigenic in our system? And so we started broad. We generally use mouse models to study pancreatic disease. Pancreatic cancer is actually made up of a lot more stroma, immune cells, and fibroblasts, a lot more scarring than it is of cancer cells. They're actually the minority of disease. So it's very difficult to study pancreatic cancer without studying the interaction of every other cell type to disease progression. So we use mouse models. And they're also very good because they're relatively fast. You can get disease to form within a year, whereas in humans this can take up to 20 years. And we can control what genes are expressed. So to answer this question, we imported a mouse model that does not express the one gene necessary for tough cells to form. And we can thank the intestine people for figuring that out. So we imported that, we crossed it into our KRAS model so that disease would form, and then this is kind of just the punchline. By 6 months of age, this is my tough cell knockout. I have much more advanced disease. And by a year, I have cancer in my mice that don't have tough cells, as compared to KC mice or just the mice with just KRAS, where they just have, I'd say, higher grade lesions. Okay, so now we know these cells are anti-tumorigenic. So what do they do? They're thought to be secretory. What are they making? What are they secreting that's blocking disease? And so I went to one of our newest technologies, RNA sequencing, and what that does is we can look at every gene that's been transcribed in the cell, so what's being made. And this is actually a lot more complicated than it sounds. You'll see all kinds of literature nowadays. We sequence this, we'd sequence that. Different systems have a lot of different issues. So if you're going to look at something like the intestines, it's relatively easy to get the cells out. You can do single cell sequencing, you can do bulk sequencing. It's not too complicated. And even if you're looking at a rare cell type, there is a lot of intestines in a mouse. A lot of intestines, a lot of material to work with. Now, if you're looking at the pancreas, the first problem you run into is that there's a lot of these asner cells still there even when you have disease. They're digestive cells, so you take the pancreas out, they start to auto-digest. They break down their own RNA. So that's one problem. The second problem is it's relatively small, and tough cells are rare, so you're not getting a whole lot of cells. So this is the process I described in figuring out what a tough cell was. I have to do this every time I design an experiment. Start with an observation. Tough cells are anti-tumorigenic. Based on the literature, they express this cell surface protein. I can use that to sort them out. Sounds good. Gather information. This protein is very specific to tough cells. That's great. That'll help. Talk to an expert. Absolutely. I came into the lab being a histology and mouse modeling expert, not knowing a lot about RNA sequencing. And if we're getting only, I want to say 100 cells from a mouse, how do we get an in-depth signature from that? Well, I was lucky. The Wall laboratories actually, their expertise is in sequencing. So we used the most recent small cell number sequencing technologies to get a very good, very deep signature of what is specific to pancreatic tough cells in this context. So from only 100 cells per mouse, I was able to find what is expressed at high amounts or low amounts as compared to every other cell in the epithelium in the pancreas. So a lot of information, had to talk to a lot of people, had to modify a lot of protocols. Absolutely talk to an expert. I am not a bioinformatician, right? Now we have a lot of data to sift through. We have a lot of talented people in the Wall laboratory, so they went through my data. They built this network of what's expressed in tough cells but not non-tough cells, and we came up with a lot of lipids, which is actually a place that the field is moving to. We're finding a lot more lipid signaling in pancreas tumorigenesis. This is already a little bit in the literature, but we're really glad to see something specific come up. These are inflammatory signaling lipids, eicosanoids. They're lipid mediators that have a short half-life that can signal to immune cells and be pro or anti-tumorigenic, pro or anti-inflammatory. So we went into what is already known. We could look up which eicosanoid synthases are in tough cells and what they might do based on what they've done in other systems. And then rinse and repeat, right? So for every step. So what do tough cells make? RNA sequencing says eicosanoids. But I want to point out that to be sure of these conclusions, it's always good to use orthogonal methods, right? So if you want to be really sure about a conclusion, you use many different tests to come to the same conclusion, orthogonal methods. So what I did was I used microscopy, and I would always suggest doing this. You're familiar with architecture, form begets function. This is absolutely true in biology. What does the cell do? Well, what does the structure lend to? So those cartoon ducks that I showed you, those really simple circular ducks, this is what they actually look like. This is scanning electron microscopy. And like I said, talk to an expert. Our imaging core has some very talented electron microscopists, very talented, and I couldn't have done it without them. So this is what this lesion actually looks like here and here. And if you zoom in really close, you start to see these microvilli. So these are my tough cells. And you could see vesicles that appear to be budding from the tips. And that's consistent with a secretory cell type. And then I love this, just for fun, we have this newer technology in called serial block face scanning electron microscopy. So we take a scanning image, and then an ultramicrotome takes a very small piece from the tissue, and then it takes another image. And it repeats that. So you can generate something like this movie here. So this was really fun. So no one knows what cell types make up those ducts. And now we're going through an entire one. And this is the tough cell here. So instead of a static image, we can look through the entire cell, or the entire lesion. And then we can go through each individual image and trace out structures we're interested in. So there's the microvilli, there's the plasma membrane. Yeah, it's cool. Some tissues are pretty easy to look at, like brain. Pancreas has to be difficult. The duct, the empty space, causes flashing when it's taking a picture. So that's what this flashing is. And if it gets too hot, it can melt the tissue. And so here's a 3D rendering. So I didn't make this, but I work with really good people. And we did this because we don't know anything about this cell type. We don't know anything about this cell type in pancreatic disease. And what we found is there's a ton of lipid droplets in here, which had not been described before in tough cells in any tissue. And lipid droplets could have a number of different functions, but it really narrows down what the cells could be doing. What's interesting is that in a lot of immune cell populations, if you give them a stimulus, they will form lipid droplets. And then if you give them another stimulus, they'll secrete more eicosanoids. So the lipid droplets act as lipid stores for these inflammatory signals to be made and released. So is that true in my cell type? This is a lipid droplet stain, and this is an enzyme that makes eicosanoids. And you can see them co-localizing here and then here. So it's very possible that in the context of mutation, these cells form, they generate these huge lipid droplets as sites to make eicosanoids to control disease progression. Okay, so now we've gotten here. What do they make? All right, everything's pointing to eicosanoids, lipid signaling molecules that control the immune system. What eicosanoids are actually in the tissue?
Go back to that part where we were talking about the lipid droplets. So they were inside of the tough cells, but then there were also vesicles that you showed that were being secreted. Was that the lipids being secreted back out?
Oh, we have no idea. No idea. Tough cells, I'm only showing a snippet here, but they express antibacterial proteins, antivirals, all kinds of cytokines. They're actually a lot more similar to immune cells than any kind of barrier epithelial cell. What I think is that they secrete these things based on the stimulus that's present. We also don't know how they secrete. There's a lot of good work that's come out of Vanderbilt showing that just the normal cells in the intestine, the microvilli act as secretory organs. And there's actually digestive enzymes and little vesicles that come off the microvilli. So I suspect it's something similar, but what's in the vesicles is more specific to this cell type.
So in your previous slide, it said that the tough cells were pro-tumorigenic?
Oh, did I not change that?
I just wanted to make sure that...
I had to go back through because I wrote, it's supposed to be anti.
Okay, good.
I thought I went through and changed it, but maybe not in this version.
Okay.
Deleting them is pro-tumorigenic.
Yeah.
That's what I did when I was making this. It's anti. Sorry. Okay. So what eicosanoids are actually in the tissue, right? Because what could they be making? The enzymes that make eicosanoids have this massive cascade that can make all kinds of different signaling molecules. So we took whole pieces of tissue between normal pancreas or pancreas that had tough cells, and we sent it for mass spectrometry to see what eicosanoids are there. And we just, as compared to normal pancreas, had 10 tons of this eicosanoid called PGD2. So then if you go to the literature, PGD2 is suppressive. It's known to function in tissue healing. And there's just a ton of it in pre-invasive disease. Do tough cells make PGD2? To answer this question, we went back to our RNA sequencing and our microscopy. And these are static, so we can show that tough cells express the genes necessary to make PGD2. This is the synthase responsible. It's much, much higher in tough cells than other cells. And this is the protein, the HPGDS protein. It's expressed in cells with these actin rootlets, so it's expressed in tough cells. So they definitely have the ability to make PGD2. So we have yes.
What is PGD2 again?
It is one of the eicosanoids, because there's a lot of them, but it's suppressive. So it functions in injury resolution, and it's been shown in a number of systems to be anti-tumorigenic. So that fits with our model. And I don't want to go into too much of what this exactly is, but if we want to know if tough cell-specific HPGDS or tough cell-specific PGD2 is necessary to block disease progression, what we do is we go back to our animal model and we knock out the enzyme that makes PGD2 from the epithelium. So we knock it out from tough cells. Do we get the same phenotype? And the answer is that we get a partial phenocopy. So yes, we do. Tough cells have the ability to potentially make a number of eicosanoids, so we would expect a partial phenocopy. So what you're looking at here, this is histology of mouse pancreas. This is the normal pancreas. This is a pancreas with a KRAS mutation, so some disease. This is the normal tissue. The mouse that has no HPGDS in the tough cells, much more injury, much more disease. Amylase, this marks our normal asner cells, lots of them in a normal pancreas, far less in the mouse that lacks HPGDS than the mouse that has it. And then this is a fibroblast, much more activated stroma, much more scarring in the HPGDS knockout mouse. And that takes us here. So based on this work, the conclusions that we draw, and then there's a lot of areas now to study coming out of this work, what we found is that in response to KRAS mutation, these asner cells become ductal cells, which was known, but they're not just heterogeneous in terms of being irregular or mutated or having different signaling proteins on. They're actually different cell types, and a proportion of them are tough cells. And these tough cells react to this disease state by secreting things like PGD2 and then I think probably a number of other mediators. And this suppresses the microenvironment. So it turns off fibroblast activation, turns off immune cell activation. And disease forms, but it forms slowly. These mice don't get cancer until about a year and a half. But if you don't have tough cells, you still have this transdifferentiation event. You have a lot more activated stroma, a lot more pro-inflammatory signaling, and cancer forms a lot faster. So in conclusion, I started with what is it, then I said what does it do, and then we came to this unknown signaling pathway in pancreas. We also learned a lot about cell state plasticity, cell state switching, and previously it was thought that these metaplastic and neoplastic lesions were there to seed cancer. Like you find them there, that's bad, they can progress to cancer. But what we're finding here is that these lesions form to protect you. A number of these cell types are derived from the normal tissue to protect you from the disease getting worse. This is a new mechanism for the body to stop disease before it can progress. So if we research these cells and we figure out what they make, can we mimic that as treatment? If we now know they're there and that they secrete specific substances, can we check the blood and know that there's a problem in the pancreas? And those are the directions that we're taking this research. So in conclusion, I go through this, daily for every little question that we have to ask. So it's good to keep in mind, and it's great to talk to people. I always say to talk to people. And like I said, talk to people. A lot of people help me with this. This is our laboratory, my mentor, Jeffrey Wall. I have a number of Razi and Vikas technicians that I train and work with. They've become experts in a lot of this sequencing and histology. Vikas is an undergraduate student. Razia had a master's degree from a great institution and then stayed home for 10 years to have 4 kids. And she came back to work a year ago, and she wants to go to graduate school, so she's been working really hard. We're getting her on a paper as co-first author, and then we're working on her application. Bioinformatics, people with pancreas expertise, people who help treat the mice. Our cores, like I said, our microscopists, I love working with them. And then a lot of collaborators and funding sources. So if anyone has any other questions.